interesting species. Okay? And it is photosynthesis and plant productivity. There are two different approaches we have taken, but due to shortage of time, I will only skip one. But before I go, I must say this. But these are chlorophyll biosynthesis pathway here. One is synthetic pathway, and another is degradation pathway. What I am saying here, that chlorophyll biosynthesis and degradation pathway are also interconnected. Okay, and we are modulating one of the enzymes of chlorophyll biosynthesis. Here I have said two enzymes were modulated, but here I am talking about one enzyme today. We modulated chlorophyll biosynthesis pathway. So I will skip one because we don't have time for that. So I will just go to one of the enzymes we have modulated to increase photosynthesis and plant productivity. This is Protochlorophyllide oxidoreductase. You see, plants have, an enzyme, photoenzyme called Protochlorophyllide oxidoreductase. Its function is to convert protochlorophyll to chlorophyll during chlorophyll synthesis. When seedling grows, germinates and grows, massive amounts of chlorophylls are synthesized in the seedling. Okay, otherwise in response to light. But you see that this I talk about, I don't know. This is chlorophyll biosynthesis pathway in nutshell. Glutamic acid goes right to the delta aminolevulinic acid, give rise to protoporphyrin 9. There is magnesium inserted, it goes to chlorophyll, but if iron inserted, it goes to heme and cytochromes. The plants, both the pathways operating, magnesium pathway and iron pathway, both are operating, while the animals, the magnesium branch is shut down. Okay. So we have protoporphyrin 9, magnesium inserted called magnesium protoporphyrin 9. Then you have protochlorophyllide. So finally this is a protochlorophyllide. Then protochlorophyllide is converted to chlorophyllide by the enzyme in pencil light by the enzyme protochlorophyllide oxidoreductase called POR enzyme. Okay. POR enzyme is a light dependent enzyme. It needs light as an obligate requirement for synthesis of chlorophyll. And once chlorophyllide is formed, it is phytylated to form chlorophyll. Okay. So this is a nutshell the chlorophyll pathway. I am talking about one of the enzymes of the nature, is the protochlorophyllide. There is another enzyme, also widely known enzyme, which needs light as a substrate for its action, the DNA photolyase, which is needed for DNA repair. Okay, and pyrimidine dimer repair in UV light repair, and I am not talking about that today because it is not present in higher plants either. So, okay. So we are talking about this enzyme here, which is light enzyme. Okay, photoenzyme. If you see in the process of evolution, this is protochlorophyllide structure, and this is chlorophyllide. What is happening here, the double bond here is reduced to single bond here. There is a difference between protochlorophyllide and chlorophyllide. Okay. And this enzyme, protochlorophyllide oxidoreductase, converts protochlorophyllide to chlorophyllide. And once chlorophyllide is formed, then phytyl tail is attached here to form chlorophyll. Alright. So this protochlorophyllide can be converted to chlorophyllide by light dependent enzyme and light independent enzymes. If you go to algae fungi in algae, bryophytes, pteridophytes, and gymnosperms, pines and all. They can green in dark. Okay, they can synthesize chlorophyll in dark because they have light independent pathway, light independent chlorophyll biosynthesis pathway, light independent enzyme. Okay, these light independent enzymes have 3 subunits: CHL, CHLN, and CHLB, and these 3 genes code for a light independent protochlorophyllide oxidoreductase, a light independent pore, and these are present in chloroplast genome. Okay. So, in the process of evolution, these 3 genes have been deleted from angiosperm genome. So, angiosperm chloroplasts do not have these 3 genes. Okay. Therefore, angiosperms cannot green in dark. Okay. They need these 3. They are only have photoenzyme, the light dependent enzyme, photoenzyme, and we have 3 isoforms: pore A, pore B, and pore C. Pore stands for Protochlorophyllide oxidoreductase, the enzyme that I am focusing today. Okay, synthesize chlorophylls. Okay. If you grow a plant in dark, plant shielding is yellow, not green. That is because plants do not have, angiosperms do not have these 3 genes in their chloroplast genome. Okay. All right. So when I say pore A, pore B, pore C genes, there, so there are, in Arabidopsis there are 3 genes: pore A, pore B, and pore C, and their similarity is very high. You can see how much they spare. Pore A is 1 to 1.8 base pairs, pore B 1 to 0.6 base pairs, pore C 1 to 0.6 base pairs. You can show significant relationship by this, all happened by gene duplication events, but this pore has, but they are similar ends. Okay. So while looking at the Arabidopsis genome, we came across a gene which is called pore C, and we put this genome, this protein, this gene in E. coli, expressed in E. coli, and found out, yes, it's similar to pore A, pore B, but yes, it can also synthesize chlorophyll in light. So what we did, we expressed in bacteria, cloned this, expressed purified in nickel column, and the protein. Then we took a cuvette, fluorescent cuvette, added protochlorophyllide oxidoreductase, isolated protochlorophylline from the seedlings, put them together, okay, and this is the absorption fluorescent spectrum of the substance. This is protochlorophyllide fluorescent spectrum at 635 nanometer, and as you see shine light, you can see here the protochlorophylline fluorescence goes down and chlorophyll fluorescence comes up. Okay. Okay. So from this experiment, we proved that this enzyme is really can convert protochlorophylline to chlorophyllide. Okay. So this is a C isoform of protochlorophyllide reductase. We call it pore C. It is present in Arabidopsis and most brassicas, including Brassicadentia and many other crops. Staining experiment, we isolated protoplasts from wild type and transgenic plants, and so is the staining to measure the green fluorescence measured by induced by single oxygen, and you can see the protoplasts isolated from wild type plants without ALA, with ALA, the Porcix plants, and the mutant. So you can see here the single oxygen production is very high. The fluorescence you can see for wild type and with the ALA you can see the chloroplasts are disorganized, and you can see a lot of chlorophyll fluorescence, single oxygen production. For Porcix plants, we have seen very low. But if you give Porcix to mutant plant, you can see where no protochlorophylline present, you can see they are dying very, very fast. Okay? And also, we saw the analysis showing that the programmed cell death, the single oxygen produced in the chloroplast is able to execute the cell death in the nucleus. Okay? And therefore, the programmed cell death executed in the nucleus by analysis, so that therefore what you're saying is retrograde signaling by the single oxygen which killed the plant, killed the cell through some through necrosis and some through programmed cell death, PCD. All right, this is basic. So we just go ahead a little bit further and stop saying, yeah. These are your wild type plant. Yep. Wild type control plants, Porcix control plants. You can see a clear difference between this. And we gave them stress, short stress, after a few days of stress. And after 2 weeks of stress, you can see wild type control, Porcix control. They are doing so good, but you give wild type stress and Porcix stress, you can see why there is no flowering took place. In wild type plants, or Porcix plants are gone and flowering. And also this electric stages of growth, and we measure the fluorescence, electron transport rate. You can see wild type transient, you can see clear cut increase in photosynthetic rate. Okay, and you can see the stress condition with the wild type plant is very, very minimal. Photosynthesis is more. Yeah. We also measured the CO2 assimilation rate by the leaves. You can see the wild type and transgenic, showing the photosynthetic capacity of the plants are increased. Okay, not only its photosynthetic capacity increased, but also it is resistant to short stress. As you can see here, the short stress have changed. Like quantum is photosynthesis. So, and okay, this is the dry weight, this is the control. You can see here, control wild Porcix is always red, wild type is always blue. So you can see wild type Porcix overexpressors, and control plants always they have dry weight higher, 150 millimolar NaCl, 200 millimolar NaCl, showing that the wild type plants are being, transient plants are being better even stress conditions. And starch content you can see also much more in transgenics, even 200 millimolar NaCl. And this is the weight of the seeds per plant. You can see the yield also has reduced, but not as much as in wild type. 1,000 seed weight is also much higher in the transgenic than wild type. So I come to conclusion here. Attempts are made reducing the oxygen concentration of plant cell under control stress conditions. I will skip this portion here. Overexpression of Protochlorophyllide, C, efficiently phototransformed, Protochlorophyllide to chlorophyll, leading to reduced accumulation of photosensitizer, Protochlorophyllide, and minimal production of oxygen. Reduce accumulation of oxygen, protected plant oxygen phase, and increase plant productivity. Thank you very much for paying attention.
So any questions for sir?
Yes. No. Yes.
I heard you talk before as well, and nice work. I have two questions. Although you showed with the Porcix overexpression, it's increased the chlorophyll contents, and then finally under stress condition it was salt tolerance. And
yes.
So have you tested this under different stress conditions, because chlorophyll increased photosynthetic efficiency will be affecting will be under other stresses like drought and high temperature and others, it will be much better.
We tested drought. Now we are testing high temperature. We have already done drought experiment, which I am
Okay.
not showing here. But drought also, there is drought tolerance to a certain extent. But we feel we see better high temperature tolerance.
Okay. And my second question is because the glutamate is key to the synthesis of the chlorophyll. So under this, when you overexpress this gene, you are basically pulling more glutamate. So the carbon-nitrogen partitioning will be affected. If the plants don't have enough nitrogen fertilizers, then the glutamate will be limiting. So how was the carbon-nitrogen partitioning? Have you tested those in these plants?
Yeah, you see, only if there is nitrogen limitation in the soil, then the probability may arise. But under our experimental conditions, where there are no nitrogen limitation in the soil, or we did a pot experiments, also did soil experiments also later on, okay, on the field with special permission. We saw that they are doing much better. And not only that, they have also small increase their protein content also.
But because most of the time the farmers has to, the nitrogen will be limiting. Farmer has to, they have to put the fertilizers.
Yes.
And if in this scenario, it is pulling more nitrogen towards this photosynthesis. So that will be.
It is side change, you know. You see how much chlorophyll is there compared to protein. Much, there much more protein and very, very less chlorophyll.
Okay.
Milligram to microgram
Yeah.
situation. So in that case, we don't expect much protein to be transmitted to chlorophyll.
Okay. Yeah.
Okay.
Thank you. Yeah.
Anyone else?